So thank you all so much for joining us today for our Knowledge Portal focus group. This is the first of the year for 2026, and we are very excited to have a wonderful new year with all of you. And as part of that, one thing that I want to highlight is in addition to our social media that we already have for our LinkedIn and Twitter and things like that, we also are now on Blue Sky. So if you have a Blue Sky account, please feel free to follow these. There's, I think, a couple other that I've created as well since then. But we want to interact with all of you in as many ways as possible. So please feel free to join us on there. And one thing that I just wanted to highlight briefly was this is the schedule for 2026. So we want to bring this to your attention just so we can start, planning for all of these sessions. So if you are thinking already ahead, please hold these dates for our future meetings. And so with that, those are my basic updates. I will now hand it over to Noel, who is going to be kicking off this focus group. And one other thing is please feel free to raise your hand, ask questions throughout the session. We will also be using the chat, so hopefully everyone has that available for them as well. So I will hand it over to you, Noel.
Excellent. Thanks, Mackenzie. Happy New Year, everyone. It's great to see everybody again. And I actually want to, be a little honest with you. It was funny. We had a hole in the speaking session for the focus group, and Ryan, who you're going to hear from, had some hot off the press results. So he came to our rescue, and we've put this together for you because I think it's actually incredibly exciting for it to be a New Year's new set of results to share with you because it's very cool results that I think validate an approach that we've had on the Knowledge Center for CFD for quite some time now, but we haven't been able to put, real muscle behind it and say why we think it actually is useful. So you're going to hear a little bit about that today. But for those of you who haven't even heard about what this is and why this matters, I'm going to start off with a little bit of that. So let me share my screen and just give you a little bit of orientation before Ryan and Julie dive in. So there we go. Right? Does that sound, can you guys
Yes,
see
that
my
looks great.
screen okay?
Mhm.
Excellent. All right. So today's about serving a new research community. And we are card-carrying human geneticists who build resources. That's what we do really well. Or at least we hope we do really well. And we also think as human geneticists, we want to make genetics data more useful to others. That's kind of like the core tenet of what we do with the resources we build. And so to do that, we work with geneticists to help us do that, but we also have expanded over time to work to other communities that have other types of data that they generate or who translate what we learn from human genetics into experimental validation, drug targets, or who actually don't even think about genetics, just have a process they study. And genetics is sort of like an add-on as they are, writing up a paper or thinking about as something, if they put forth a hypothesis, it adds support. So we think about those communities and we want to get, better and intercalated with those communities. And we've done this through new resources that we've started to build. So today's focus is about the Common Fund data ecosystem. I'm imagining many of you on the phone have heard about this, but if you haven't, I'm going to spend a little bit of time talking about what it is and why it matters, because it's sort of an anchor to some of the results you're going to hear today and the data that we're pulling from. And I think that's important to just convey. But remember, we're human geneticists. The other big data ecosystem that you're going to hear about is all of the human genetics data that we work with, right? We build this association to function knowledge portal, which has 1,300 traits for human genetics linked to disease and calibrated to traits that are community driven, right? So this is the things you hear about us talk about in this webinar often. But this serves human geneticists really well. What about folks who study mouse models, right? What about folks who just are working on a specific gene and actually want to see what the process and how that gene affects different cell types that they're investigating and so on and so forth. So with that, we've branched out into another set of things, which is this Common Fund data ecosystem. And I'm going to talk a little bit about that. But the resource we built for that's going to allow you to navigate and where you're going to hear Julie talk about how to find the resource that we're going to talk about the results from later is on this site here. It's cfdknowledge.org. If you haven't used it, check it out. But this is to organize everything that is CFD. So what is CFD, right? CFD is pretty audacious and bold. It is something that the NIH has been working on for the Common Fund for many years now, but has tried to bring together things inside the Common Fund. So the Common Fund is a set of different programs. Really, that is a vision from NIH to create catalytic resources or to transform the workplace, ironically, and then also to create transformational science and discovery. So you can see the different programs that you probably recognize that fall in this broad umbrella of NIH-funded Common Fund mechanism activities. So you got everything from MotorPack, which you might have heard from, to xRNA, to Spark for these big translational programs, to things like HubMap, GTEx, Kids First, metabolomics, to these other things which you're probably not as familiar with, but like complementary, which is actually focusing on to use NAMs or new approach methodologies to study disease, right? So these are different types of big programs. They're multiple years. They're about generating data. They're all open access. The Common Fund data ecosystem is meant to sort of bring together some of them into an ecosystem. So not every single Common Fund program is in here, but many of them are. There are about 18 data coordinating centers, they're called, who are generating data. They're repositories who have these beautiful consortiums around them of scientists who are contributing, generating data, providing scientific insight and guidance, studying diseases, processes, traits, you get it, you name it, in this environment. Then there's centers at the center of it, to help coordinate, make the data accessible, train users in all of these different things, and pull out knowledge. And also, there's also a cloud component which allows you to run compute on these. So all these centers and coordinating resources are meant to work together as an ecosystem. That's a lot. We don't expect everyone to know everything because everyone has domain expertise, but the goal of the data CFDE is to really accelerate discovery, right? So because these resources are all catalytic and they all have their own environment, how would you make useful of them? So what we hope the Common Fund data ecosystem does is to allow query and cross use across all the data sets that I showed you in that beautiful diagram to sustain them, meaning not necessarily to make sure that, these things are funded forever, but the results, the really important things that come from these various data coordinating centers that I mentioned, after that they might sunset from funding. How do you sustain their results, the knowledge? Obviously you have publications, but you really want to make sure, like if someone wants to use that data in future, how do they find it? How do they make sense of it? How does it matter to them, right? That's our job at the CFD. And to train folks in using it. That's kind of the core. But when you saw that thing, you probably looked at like how would Glygen, who studies glycosylation, have anything to do with the druggable genome or to bridge to AI? Well, they have a lot to do with each other. They have themes and threads. They have things that are in common, right? They're all data aggregators. They tend to all be reference maps that create these open access reference maps of whatever they're generating. And they link to molecular signatures. So those are these themes and thread that run throughout the program. They also have core values. Everything's fair, which I can just guarantee you, having worked with them for 3 years, it's pretty impressive how these resources build. It's pretty cool. It's taught us a lot about how we build resources. They all have their own communities that they serve, right? You have a project that's called Senet that just works with senescence. So they work with aging, how cells age, right? They have a community around that that's deeply engaged in that research. They engage with the external community to educate, train, share the results. So that is a core tenet, and that's also something that we as a center share. So our job together is to make CFDA data more useful and enable discovery to a wider set of community resources. So we can't do this all at once. We try and do it carefully, but we're human geneticists, don't forget that. So when we approached this project, we thought as the Knowledge Center, we need to enable these resources to make their results more useful to a wider community. How best can we do it? Well, we understand human genetics. We understand how, variation in your DNA can link to a phenotype through a statistical measurement and what that says potentially about biology. Well, what's great about that is if you can learn that link, you can also connect all of the CFD data to that genetic assertion, right? That association. So our job at the Knowledge Center is to take the best of, what we know about human genetics and marry it with all of the data and the results in the CFD. So that's what you see on the Knowledge Center here. That's kind of what we built it as our primary initial offering for the resource. You can learn about all the programs. You can navigate through the centers. I welcome you to do that, but that's not the focus of today. What the focus of today is we have specific workflows that are geared towards different users. We have folks who want to think about genetic analysis and gene sets. That's one module that we're not talking about today. What we are talking about today is human geneticists and mouse geneticists. So the module you're going to hear about today is the mouse to human phenotype analysis. So what this allows you to do is to match phenotypes according to genetic similarity from the mouse to the human. And we think this is very helpful because this gets done, this is how it works, but this gets done typically by semantic search. And this is really, valuable in the field. And I know folks that are mouse geneticists who know way more about this than I do will understand this and be able to describe this better than I can. But this is how this is typically done, right? They look at semantic similarity. They observe a phenotype in a mouse. They say, what is the semantic match to humans through the ortholog? And they do that research there to help them translate through that to the human phenotype. And this is done through things like the MGI resource and so on and so forth. But the challenge with that is, right, we need to make sure that we're all trying to find better disease targets. And Ryan is going to, expand on this much better than I. But the idea is that we want to make it so that if you're doing work in a mouse, you want to make it so that you could, ideally, you want to treat a human with a drug target, right? Often, phenotypic similarity does not imply genetic similarity, and genetics we know human genetic support does increase the relative success of something being a drug target. So with those assumptions, we want to use that with our genetic background that we've built through the association of function knowledge portal to strengthen the likelihood of the mapping of an observed mouse phenotype to a human phenotype and make that more useful to others. And so we're using this with these two components, our human genetics and the data set, the CFD data ecosystem as the two core features to pull this together into one resource. So with that, you're going to hear from Ryan about why and how, and how we've calibrated and validated that result. And then Julie's going to walk you through the resource so you can actually see how to use it, why it matters, and what scores matter and why they would be useful to you. So with that, I will turn it over to Ryan.
Thanks, Noel. Hello, I'm Ryan Koesterer. I'm going to talk today about metrics for prioritizing drug targets. A few years ago, these three large organizations, Bio, Informa Pharma Intelligence, and QLS Advisors, published a report about the clinical
Ryan,
development.
do you want to share your screen?
Oh, did I not do that yet? I'm sorry.
That's okay. I do it all the time. Trust me.
Sorry. Okay, there we go.
No worries.
Do we all see it?
Just give it a second. There it is. You're good to go.
There it is. Okay. So this diagram is shows how drug candidates sort of go through the system from phase 1 through approval. And this journey takes approximately 10.5 years. This is data based on the 9-year period between 2011 and 2020. And only about 7.9% of drugs that start clinical trials gain approval in the end. And this is a problem because at every phase of the process, there seems to be a lot of attrition. And it's interesting that it varies a little bit by disease type. So rare diseases seem to go through to approval a little more often than high prevalence diseases. One possible reason for this was posited by Sook et al. In 2013. And it sort of exposes a translational gap from mouse to human. So if you look at this grid, it describes changes in gene expression and blood immune cells over time following an inflammatory event in human and mouse orthologs. So for example, if you look at the human burn versus human trauma, you can see that the gene expression over time seems highly correlated, and it definitely is at 0.91 R squared. But if you look down at human versus mouse, you can see that there's very little correlation there. And also, as you even look at mouse trauma versus other mouse trauma, there's very little correlation there. Sorry, I'm going to move something. So why does this inflammatory response differ in mice than it does in human, as opposed to humans? They presented evolutionary distance as a possibility. The simplicity of mouse models in that they're often single mechanism oriented. Inbreeding in mice versus heterogeneous humans. The trajectories for humans tend to be shaped by things like care, fluids, drugs, surgery. And humans tend to be far more sensitive to inflammatory challenges, so there may be an upper limit to how they respond to them. So in 2025, the NIH announced funding cuts to align with their initiative to prioritize human-based research. So along with this, we see a big rise in NAMs, which are new approach methodologies. And it basically is, informally referred to as non-animal models. These are useful because they're more direct human relevant, and they're a little easier to isolate the mechanisms. They have rapid iteration. It's easier to control them. The outputs tend to be computation ready, so much easier to analyze the results. But animal models are still quite useful, especially when we need to look at the whole organism. So if we're looking at things like multi-organ interactions, overall physiology, things like absorption, distribution, metabolism, and excretion studies. Also, things like complex endpoints like neurobehavior and chronic disease progression. These all require a more comprehensive approach using animal models. So if we think about how mouse model selection is done, the sort of standard workflow is to choose a mouse phenotype that resembles the human trait, perturb the genes, and then nominate targets. And it often leans on things like semantics, ontologies. Does this phenotype in the human look similar to the phenotype in the mouse? But I want to focus specifically on the part of finding similar mouse phenotypes. So one way we can do that is by semantic similarity, and we have a way of quantifying that so that we can see how alike human phenotypes are to mouse phenotypes. The way that we did it was to use a high-quality sentence transformer model, which takes a human phenotype and converts it, or a mouse phenotype, and converts it to a vector of dimension 768. It's a high-quality model that's available freely and widely used. And then what we can do is when we have our vector embeddings of our phenotypes, we can compare them in mouse versus human phenotypes. And the way that we compare them, one way to compare them is to use cosine similarity, which is essentially the dot product of the two vectors. And so a value of, and it's normalized, so a value of 1 means that the vectors are pointing in the same direction, so they're very similar semantically. And a value of 0 means the vectors are orthogonal, so they're pretty well unrelated semantically. All right, so now that we have a way of selecting our mouse phenotypes, we need a way to go from mouse phenotype to human gene so that we can elect targets. So the way that we did this was taking two sources of genotype-phenotype assertions. So we have MGI and IMPC. They're both large organizations that collect genotype, knockout mouse model results, things like that. MGI is broader in scope of the genotypes that they use, and it tends to be published mouse models that are not really based on standardized pipelines. It's a curated assertion style, so there's not really a lot of statistical data that it's based on. The IMPC is uses standardized pipelines, and it's grounded in statistics, so it has a much more uniform experimental context. So that gets us from mouse phenotypes to mouse genes, and then we need to go from mouse genes to human genes. So two sources that we use for ortholog mappings were MGI. They publish an ortholog mapping, and also Alliance of Genome Resources publishes one as well. So the table at the bottom just shows the results of all of the mappings that we were able to do from the mouse phenotype to the human gene. and is split up by MGI, IMPC. And IMPC is sort of an umbrella organization that brings in outside data. So there's 5 different sources of that data as well. So as you can see, in this case, we're going to use the one-to-one mappings because it's a large number of them and it's almost all of them. So, and it would be easier to work with as opposed to trying to figure out all the one-to-many relationships. Okay, so now that we have a way of selecting our genes, maybe we can use the semantic similarity between mouse and human phenotypes as a metric to estimate the plausibility of target indication pairs. So we propose this baseline model where we first compute semantic similarity between human phenotype and every mouse phenotype. And then for each gene, we collect all the mouse phenotypes where G is implicated, and we assign a score as the maximum of the similarity between the human phenotype and that mouse phenotype. So now that we have our way of defining our score and our metric, we're wondering how can we evaluate whether this score has any plausibility or it can be used as a way of determining plausibility of a target indication pair going through and being successful in a trial. Minikel et al. Published a paper in 2024 that introduced a method called relative success. And it allows us to translate our semantic-based plausibility score into a clinically relevant one by asking the question, how much more likely are high-scoring target indication pairs to progress or be approved compared to the lower-scoring ones? So the mechanism here is for a given threshold tau, we call the target indication pair supported if the semantic similarity score is greater than or equal to tau. Otherwise, it's unsupported. And the relative success is calculated as the probability of all successes given supported divided by the probability of success given unsupported. So if we can say that if RS increases with tau, the score is a useful prioritization signal. So we did that. We evaluated relative success given varying thresholds of our semantic similarity score metric, and we had almost 2 million target indication pairs. And you can see in this plot, the percentile on the bottom or on the x-axis and the RS score is on the y-axis. And there's a secondary y-axis that is the number of target indication pairs included at that threshold, and that is the dotted line. And as you can see here, as the threshold increases and we get less and less target indication pairs being put forward, there's not really any notable increase in RS score. So this is probably not a useful metric for prioritizing target indications pairs. So we wanted to sort of put some genetics rigor to this and use a more genetic-driven score. So one of the approaches was already developed and is available on the portal now, or the results of it are available on the portal, is Pigeon, and this systematically integrates experimental data with human genetic evidence so that we can calculate a score that tells you if your mouse signature is more strongly aligned with the human disease's genetic architecture or not. So basically, this is a new metric that we can use, similar to the way we use the old one. And we also put that one through our test, and we evaluated different thresholds for beta uncorrected in almost 1.5 million target indication pairs. And you can see here that as the threshold increases, there's a clear improvement in the RS score. And it indicates that the beta uncorrected is probably a good metric for prioritizing target indication pairs. You can see in the top point, the beta uncorrected is 2.91. The RS score is almost 2.37. So that means the probability of success is 2.37 times more likely if you use this threshold. So yeah, so these beta uncorrected scores are available on a public platform that Julie is going to present to you next. Thanks.
All right, thank you so much, Ryan. That was fantastic. So I'm going to share my slides. Give me just a second.
Hey, Julie, while we're doing that, while we pause transition, any questions for Ryan or thoughts or counterpoints or? All right, that's okay with me. Let's keep going.
If you have
All
any
right.
Obviously,
questions
tap them in the
throughout,
chat.
just please feel free to use the chat if you don't have them now.
Yep. Thanks, Mackenzie. That sounds great. All right, so thank you to Ryan. As you've heard, this is a new method, hot off the press. It's extremely exciting because it shows promise for improving relative success score measurement and therefore improved, clinical trial applicability. So what I'm going to tell you about now is how we've translated the method that Ryan just described to you into a publicly accessible platform that you can access today and test out for yourself. So this is available through the Common Fund Data Ecosystem, or CFDE, through a feature that we call our Mouse to Human portal. So Noel told you already about what the Common Fund Data Ecosystem is. But the benefit of this module is that it helps address the problem that Ryan referred to previously, which is that these preclinical trials are often conducted in animal models, but the findings from those animal models are often not translated to humans within clinical trials. So we have a lot of sort of waste in this pipeline in getting from things that are applicable to animals into actual targeting drugs that are applicable to human health. Um, One of the reasons for this, as Ryan said, is because phenotypic similarity does not necessarily imply genetic similarity. And the sort of gold standard for the field to date, as Ryan mentioned, is what we call semantic similarity. So the idea that, a term might sound similar to another term could mean that those two terms map. What we've developed is this mouse to human phenotype module through the Common Fund Data Ecosystem Knowledge Center that enables you to match phenotypes according to genetic similarity to improve target prediction. So the underlying backbone of this is the method called Pigeon, priors inferred from gene annotations, which Ryan told you a little bit about. But the crux of this is that it is a method that can estimate the probability that a gene is involved in disease. And it does this using two lines of support. The first is GWAS support, genome-wide association studies, and the second is gene set support. So gene sets can be, any set of genes obtained from essentially any experiment. In this simplistic example, you can see that human GWAS have implicated the FTO locus in increased body mass index through multiple independent studies. But also you can see that there are sets of genes that have been identified as being linked to increased body mass index through other models, say in mouse. Those can independently implicate increased FTO expression in increased BMI. And collectively, these two lines of evidence show that there's strong support for the involvement of the FTO locus in body mass index as a human trait. Many of the gene sets that were obtained for this analysis were generated by Avi Mayan and his team at the CFDE Data Resource Center. So you can contact Avi to learn more about these tools or also access them through the CFDE Data Resource Center's site. Pigeon is also useful because it integrates nearly 7,000 multi-source common and rare disease traits. So these are obtained from our Association to Function Knowledge portals, the GWAS catalog. Both of these address common disease, and also from Orphanet for rare disease. So how does this actually work in the context of the mouse to human phenotype module specifically? So in this case, we have input data, which is provided as gene sets that are related to mouse phenotypes. These are derived from IMPC, the International Mouse Phenotyping Consortium, and the Mouse Genome Informatics Tool, MGI, and MSigDB. And we also input those common and rare disease human phenotypes from the various sources that I just described to you. We use the pigeon method to compute scores that then tell you which mouse phenotypes are related to human phenotypes. And we're able to do that by using the gene sets related to a mouse phenotype as a proxy for that mouse phenotype. So on this tool, you can query by a mouse phenotype, for instance, head bobbing here, and obtain the top 20 human phenotypes that are genetically linked to that queried mouse phenotype. Conversely, you can see a query by human phenotype to get a list of the top mouse phenotypes that are genetically related to that human trait. Within the interface, you see sort of a couple of major outputs. So for each mouse phenotype, you'll see an estimate of the probability that the genes associated with that mouse phenotype related gene set are involved in human disease. And these estimates are available for both the gene set in isolation. We call that the marginal effect. That's the same as the beta uncorrected score that Ryan just referred to in his slides. And they're also available within a joint model, and that's considering other gene sets to correct for them. So here's an example of something, an interesting biological finding that came out of this interface. This shows the power of the interface to leverage human genetics in order to find sort of unexpected phenotype connections that might not be available at the surface. So what we've done here is queried a rare human eye movement disorder called congenital fibrosis of the extraocular muscles to see which mouse phenotypes are genetically linked to that human phenotype. And interestingly, we see that some of the top hits are things that we would expect, right? They're things associated with extraocular muscle and the nerves that innervate them. But then other phenotypes are not as directly associated. There are other sort of brain malformations, such as abnormal basal ganglion morphology. If you know something about these disorders, you know that this finding can actually be seen in some forms of the human disorder and also the mouse disorder. Experienced neuroradiologists know to sort of assess for this, but others might not. So this is just one example of something that people who understand the disorder might know, but people who do not, this could give them some insight into additional related phenotypes that they could assess for based on the shared genetics. So in summary, this mouse browser provides a genetics-first approach to enable cross-species discovery. It can uncover genetic links between species, even when the phenotypes appear to be totally different. So we can therefore use it to identify novel phenotype associations. By using gene sets, we can link to a variety of other programs from both CFDE and outside of CFDE, and to other data types. So this resource is actually a model for advancing data accessibility and interoperability. And I'll note that there's a caveat here as well. So this is a brand new method. We're sharing this with you early so that we can get your feedback and continue to validate the method in order to demonstrate its utility and its limitations fully. So related to that, we would love to hear more from all of you. So in your own research, how would you actually use this interface and how would you use this method and these results? How can we improve the tool that we've developed to better suit your needs? We are especially interested in finding collaborators who study mouse to learn about interesting positive and negative controls that you've identified in your own research. So examples where you would expect semantic similarity, would expect a linkage, I guess, between a mouse phenotype and a human phenotype, but traditional methods like semantic similarity do perform well or do not perform well. And we'd love to see how our CFDE tool compares and what your take is on that. We also want to work with people more generally to understand what types of tools and resources would be useful to advance your research. We're developing new methods all the time, so we'd love to work with you. And then finally, we'd love to learn how CFDE can become more accessible and useful to you. So huge thank you to the amazing team who helped to make all this possible. Brian's done a lot of the work, but many other members of our team and of our colleagues' teams in Kyle Galton's lab at the Data Resource Center and others have helped to make this work possible. So thank you to everyone, and we're happy to take questions.
Thank you, Ryan and Julie. Since we ended early, which is not bad, I like it. We have time for questions. We also have time if you have a favorite phenotype, or if you are a mouse researcher and you study a process and you want us to kind of do things on in real time right now. We're happy to do that. That might be worthwhile doing if people want to serve as a guinea pig to say, hey, can you walk me through something, a phenotype I care about, and see what the results are? We could do that in real time. That would be valuable.
So we have a question from James Fleet. What is the link? This is just the link. Sorry. What is the link to the Mouse and Human portal? So I had put it up in the chat right before you had just put it, but I can put it back in. Let me just copy that and paste it again.
Actually, what else you can do? I mean, Mackenzie, actually, why don't you share your screen? And we could just like literally navigate folks to it. Julie kind of did this, but let's do it in real time, yeah?
Hang on, we have a couple questions.
Or Julie, you can do that, whoever can get to it faster. Yeah, you can manage the questions, Mackenzie. I don't want to get it.
Okay, yeah.
Yeah, sure. Give me just a second to.
Okay, then Nora's hand went up first. So Nora, please
Yeah.
feel free to ask.
Yeah. Hi guys, this is really interesting. So I actually go to the reverse. I do humans and then I like to look at mouse to see if they're the same. So I just wonder if there's applications for, I mean, you guys touch a little on that on pharmacogenetics and if you have any examples there.
Your question is about
Yeah.
pharmacogenetics, and help me
Yeah,
understand what you're
so
thinking.
So I was just thinking like, some say we specific in some genes, if we can actually look if there's been um checking mouse models any, in terms of experiments related to drugs or or just trying to get like from the genes we identify,
Yep,
try to get if they are druggable or not. So in
got it.
experiments, the animal experiments, and I don't know if this feature is part of the current version.
So it's part of the reverse, I think, which is you have them actually you could do the reverse, right, Julie? Actually, we could do both. You could say I have a human phenotype or a gene that I believe has a human phenotype link, and I want to know what the mouse phenotype is that I would study that would best match that.
I
Then
see.
what we're gonna that and then the statistics that Ryan showed, the beta uncorrected, is similar to relative success as a proxy for relative success. And therefore it would say like how likely would that target potentially have the relative success historically, from the historical data, because you're comparing to history, as a drug target. So that's
Yeah.
the prediction that you've got there. So it's a not completely your question, but I think it's sort of getting at the essence of it, perhaps.
Yeah, yeah, no, that's very good. Yeah, that's great.
So maybe we can show you that. Is there a phenotype? Is there a way you want to look through it? Walk through that for you more specifically? Or is that sufficient for now?
For me, it's good. It's good.
Okay. All
Thank
right.
you.
All right.
Okay, so Dan also has his hand raised. So Dan, please feel free. Great to see you.
Hey guys, how's it going? So I wanted to share a paper that I published a few years back that's a little bit related to this.
Oh yeah.
But yeah. So mouse to human. But so this was a situation where we knew the phenotype in mice and humans that would be relevant. We were looking at bone biology. And so I was working with lab colleagues that looked at gene expression that changed with age in mouse femurs. And then we, did the RNA-seq, identified sets of genes, and then I used Magma to create gene-based scores, and I tested whether genes that change with age in mouse femurs were enriched for GWAS associations with estimated BMD from the Heal ultrasound in UK Biobank. Sure enough, it was. So this is kind of similar, I feel like, in the spirit of what you guys are doing, but we already knew the phenotype that was relevant bone. But I
Right.
was kind of thinking it seemed like bone worked really well. And
Yeah.
I wonder if that could be kind of some of your positive controls that you were kind of asking
That's
about.
exactly what I
I
was thinking
think
about.
I have a sense that that would be a good place for you guys to look for positive controls where your new method that's, sounds fantastic would probably work really well.
That's an incredibly good suggestion, Dan. I love it. I'm so glad you put this paper. This is exactly the kind
Yeah.
of thing we're looking for, particularly phenotypes. It's also interesting
Mm.
because we've also looked at like how magma is, pigeon builds off of magma, right? It's like
Yeah.
it incorporates magma into its method. So it would be really cool to see that, but more importantly, to use the phenotype you suggested. So we might follow up with you. This would be excellent. I
Yeah,
like
that
that.
sounds great. And I definitely, was I had to think about which magma model to use, like the
Mhm.
top snip model versus mean model. I went with
Right.
top snip because I thought for myself, they're in the EBMD GWAS from UK Biobank, there's some allelic heterogeneity.
Mhm.
And I wanted to just take, it's complicated, frankly, finding which SNP is probably the most important in a region. So I just
Right.
went with kind of a simple model. Let's just take the most significant. It's probably representing the strongest signal in that region. But there are some simplifying assumptions that about gene proximity
Hmm.
and SNP, and sometimes that can get the wrong answer. So
Yep.
that's an issue I don't really have a great answer for, especially in tissues like bone, where there's
Hmm.
fewer connections between genetics and gene expression.
I
So
see.
there are a number of issues that to overcome. And I took a very simplistic approach, but making that SNP to gene connection in the
Hmm.
human GWAS is a critical step.
So that's important, and that's what we're hoping that our genetics hat on, our human genetics with the pigeon resource. And then there's another method that's coming online in the portal probably this spring called Falcon, which actually incorporates trait patterns and also allows you to see incorporates even more annotations, genetic annotations in the cell type specific to enhance that score. That'd be really cool to see how, we predict the cell type of action for different traits like bone, fragility. But back to your point, I love this as an idea of actually recapitulating what you did in this paper and looking at how this actually stacks up in our particular feature that we talked about today. That's really
Yep.
cool.
Yeah, that sounds
Thank
great.
you. Thank
Mhm.
you. Other questions and comments? I see active chatter from Eva, but that's being answered by Julie, so thank you. Um, oh, the note taker keeps on giving us notes, which cracks me up. I'm not quite used to this, like note taker giving us notes. It's pretty cool. The AI world we live in. All right. Any other comments? Julie, Mackenzie, Ryan, anything else you want to cover? Yeah, Dan.
Actually, I have one more. Have you guys come across the work from Ed Marcotte
No.
at UT Austin? Let me pull up one of his papers. He's done some of this work as well a couple years ago. And I'm just trying to pull up the paper for you guys. But he did some research on trying to identify homologous phenotypes from human to other organisms using shared genetic influences.
Cool.
Let me just see if I can get it while you guys are here. Yeah,
Thank you.
okay.
That'll
So
be good to
I'm
compare.
gonna send you this link. Are you guys
Awesome.
have you guys published the pigeon method?
Not yet. They're actively working
Okay.
on it.
So
And we're close. We
good.
have it. It's on the site. There's a webinar link to it, but nothing formally published yet.
Okay, so I sent the PubMed
Oh,
link.
perfect.
Check it
Thank
out.
you.
It's the PNAS paper. By now
Yep.
it's pretty old, but it might be worth a look at thinking about some of
Nice.
the ideas.
Nice. All right, cool.
All right. Thank you
Hi, can I
guys.
have a question, please?
Yes.
So in that great example of how FTO, on one hand you have GWAS data for FTO locus, on the other hand, mouse data you have FTO expression data, and the conclusion we have is that FTO is involved in this trait. I'm just curious, how can we understand the causality? The method help us understand the causality of the gene to the disease.
Julie, you want to take that one?
So at this point, what you can determine, basically, we have two scores that are available as like sub scores within the portal. So what you can get for any given example is a score that tells you to what degree the GWAS evidence contributes, and then you can get a separate score that shows you how much that gene set evidence contributes. I think to your point, in some cases, let's say in the FTO example, we know that both the GWAS and the gene set evidence contribute, but in other cases, one might contribute more than the other. So what we have is a set of subscores which tell you the relative contributions of each of those pieces to the overall phenotype. Um, it also will tell you exactly which genes within that gene set have the evidence. So let me pull up an example real quick. Sorry, I'll just pull up the site so that you can actually see that in practice.
Okay, great.
Give me just a minute, but that's a fantastic question. Is that what you were getting at?
Yeah, just curious, from this methodology, can we get any insights on the causality?
Got it. Okay. So here's an example. So here I'm on the CFDE Knowledge Center site, and I can actually just show you quickly. This is our landing page. So if you're on cfdeknowledge.org, you can get to this mouse to human phenotype module in two ways. One is through here, where you can explore, related mouse phenotypes or related human phenotypes. And then the other is through analyses up here at the top, where you can click mouse to human. So here you can toggle between the two modules. So currently we're asking which mouse phenotypes are relevant to my human phenotype. And then if you wanted to explore the converse, you'd click here. So within this module, we can start by clicking a human phenotype. So let's say obesity, and we do search. And let's see. And down here at the bottom, it will show you what the results are. So you can see, sorry, this looks like needs to be refreshed here or something. Okay. So we go down here to the bottom, you can see which mouse phenotypes are related. And if you want to understand which genes are actually potentially causal based on the genetics and or the phenotype or the gene set evidence, you can get these scores within this nested table. So here you see genes and gene set. You click show. And you can see for this particular mouse phenotype, these are the top genes that are associated that within the gene set with that mouse phenotype. And you can see The direct support scores show you which genes have GWAS evidence. The gene set evidence scores show you which genes have gene set evidence, and then the combined scores give you sort of a summary overall using both lines of evidence. So how you can interpret these for direct and gene set scores, over 3 is typically very, very strong. And then scores like above 1 are, worth looking at. If you want more information on those scores and thresholds, you can click here within the research method tab. And that will give you, sort of 1 is nominal significance, maybe interesting to look at, and then 2 strongly suggestive, and then 3 very strong. So if you ever want to look back to learn more about where those scores are and where they're coming from, you can get that there. Does that address your question?
Yeah, great. Thank you. Thank you so much.
Thank you. Any other questions?
I have a naive question. This is Doug Keel. I was just, what is the difference between these paired phenotypes using this method versus simple rho G genetic correlation between 2 traits?
I'm not sure offhand, Doug. Ryan, do you know? If not, we can take a look and follow up with you.
I'm not sure I understand the question. Can we rephrase it in a way, or?
Okay, you want to know if 2 phenotypes are genetically correlated, and so you calculate the genetic correlation between them. So you would, I guess, you could do all the genetic correlations in the mouse and all the genetic correlations in the human, and see if they are somewhat similar. I guess you formally test whether they're correlated in both species.
Doug, are you asking for how you could use pairs of phenotypes to infer mouse to human phenotype better, or are you just asking if we use genetic correlation in the model at all? Or are you asking something different?
Well, I didn't quite follow Ryan's, I'm sorry, I just didn't quite follow the statistic that he generated, and I didn't follow the graphs that he was illustrating because I didn't quite appreciate what the axes were and what he was showing. Right. So I'm not sure of how these. I know that you use these gene sets and GWAS results to find phenotypes that are genetically similar because they're driven by overlap in the genes. But right. I didn't quite understand the mathematical model. And that's what genetic correlation, in some sense, tells you whether 2 traits are genetically correlated because they have the same genetic indicators of the trait. So I didn't quite appreciate the enhancement of this. I just didn't follow quite well. Maybe that's too big of a question, but maybe everyone else
really good.
Got it.
No, it's a good question.
Hi, Doug. It's Jason. So yes, I think you have the right intuition. The way, in essence, we are looking at whether a mouse phenotype is genetically correlated with the human phenotype. And the way we're doing that is kind of looking at the genes in the mouse that are annotated as relevant to the mouse phenotype and seeing whether they're also enriched for GWAS associations for the human phenotype. And we're doing that through a method called Pigeon that is like very robust and accurate and able to do that in a way that other methods haven't been able to before. And that is like the essence of what we're doing.
So that's underlying Ryan's. Yeah.
Right.
Yeah.
So if on Ryan's plot, so then to see whether a drug target indication pair has mouse genetic support, you kind of see whether like there's a phenotype in the mouse, right? That the human phenotype has mouse evidence, right, for a mouse phenotype that's correlated. And there's a score for that. And that x-axis is the degree to which that genetic correlation exists for the mouse phenotype. And what you're seeing is when you do the genetic correlation, when you start to get high support there, that's when you start to be able to predict drug target relative success Whereas if instead you just look at whether the phenotypes sound the same, you don't see any trend there.
Ryan, can you pull up
Yeah.
that figure? It might be worthwhile to go back through that excellent figure that shows that to
And
Doug too.
and maybe do you mind if I take a stab at answering Doug's question? Because I think I maybe have an insight. So I think if I got it correct, Doug, you're asking why not just do LD score regression and do genetic
Right.
correlation, which is the rho G. The reason is
Right.
because to do genetic correlation, you need a GWAS done in 2 traits. And we have GWAS like crazy in humans, but we don't really have GWAS in mice. We don't have the same SNP information in mice. So you can't do genetic correlation across mouse to human like we
Correct.
do from human to human.
You
Is that getting
correct.
to your question?
So what data is used in the mouse side of this beta uncorrected and
So
the
in our
cosine?
case, so what we're using, so we're, yeah, so I didn't get your name.
It's
Who?
Doug,
Doug.
Dan
Dan.
and Doug.
No, I know you,
Yeah.
Doug. So Dan, yeah. So Dan was saying. So you're correct on that, right? So like, and even if you had a mouse GWAS, you can't line up the SNPs, et cetera.
Yeah, right.
Right.
So because we can't match up SNPs, Doug, what we do is match up genes because there's lots of sources to get
Okay.
genes that are implicated in a mouse trait. So you're trading off power. You're not able to do like a polygenic global correlation, but that's what this method Pigeon was designed to be able to do, to be able to like figure out how to, I mean, so it's
Map
closer
it to a gene.
enrichment,
Yeah.
but
Yeah, that's another way
right,
of putting it.
right. So, the way that Pigeon works is it takes the genes that are recorded, and there's many sources. And one thing we're going to explore is like, it's not just whether the mouse phenotype is correlated with the human phenotype, but whether the method for determining that a gene is associated with the mouse phenotype predicts whether the gene predicts the human phenotype, right? Because you could have the mouse strain could matter, like there's like a resource like the comp from Common Fund where they did systematic screens. There's like literature reported things. So all of those things, the way that you determine a gene is associated with the mouse phenotype is also confounded in the ability to predict the human trait. And that's where the approach here is really not just a mouse phenotype. It's like how the gene was implicated in the mouse phenotype. All that's going to matter. And we're going to kind of explore whether literature curated mouse genes is higher quality than systematic screens, et cetera, et cetera.
Mhm.
But yeah, so the genes are the way, so that's the idea. And like for other people, like it's another way to look at it is just enrichment. You could think of this as, the mouse gene set, is it enriched for the human trait, but we're doing something slightly more sophisticated than just stock enrichment.
Right. Okay. So on this figure, which I didn't quite follow, the x-axis is percentile, and then the score that you calculated. I
So
didn't.
think about it
What?
like you have a target indication pair, and there's a degree to which you have evidence that the mouse gene for the target, right, when perturbed, is like associated with a mouse phenotype that matches the indication, right? So you're using the mouse data to determine whether the target, right, has a phenotype for the indication. And the red line is if you do it because the mouse phenotype semantically resembles it. The yellow is when you do it genetically. They're standardized to be ranked by percentile. So the further right you go, the more mouse support there is. So what the red line shows is that even the like target indication pairs that have the highest mouse support according to semantic similarity do not succeed more often than chance. But when you get to the extremes of the genetic similarity, you start to get, that is able to predict drug target relative success, to predict drug target success at a higher, statistically significant way.
Does that resonate?
Yep. That's
And then
helpful.
the
Thanks.
final plot and then what, I think I wasn't on the call, but what really, when you really start to get signal is when you don't just say a single mouse phenotype. So for diabetes, this target had a mouse phenotype for impaired glucose tolerance, but it had 5 mouse phenotypes. All of them kind of match diabetes and collectively when you start using these type of composite mouse phenotypes, that's when you really start to see predictive power.
Ryan, you didn't show that. Do you actually have that handy though?
You asked me not to show it, but I will if you want me to.
So like
Since we went there.
So this blue graph, so now like the green's the same as the other one, but now the blue is when you start to use composite mouse phenotypes. So like for each human phenotype, there's like multiple mouse phenotypes that if all of them are observed, the relative success gets really high.
I realize we're over one, so thanks for hanging with us. But like, we will make this available for digestion. Actually, this is great discussion and exactly what we need. So I really appreciate that because it's also the first time we've explained it outside of our own inner circle to others. So it's really cool to hear you guys give us feedback about how we're sharing and what we're communicating. But it's also nice that Dan, Doug, James, and others who are willing to serve as follow-ups to make sure that we're actually validating this and actually doing some more work to make it more make sense on our site would be really helpful. So that was great. And anyone else who has more questions, you can also email us afterwards if you find that you couldn't get anything into this conversation. Thanks, Ava. Anything else? Any other closing remarks, Mackenzie? I know we should wrap up and let people go back to their day.
Just thanks everyone. Happy New Year. And just a reminder that our next webinar will be February 12th, and we hope to see you all there. So thank you all. Thank you to our speakers, and hope everyone has a great rest of their afternoon. Bye, everyone.
Thank you, everyone.
Bye.
This is nice. Take
Thank
care.
you. Bye.